Great, thank you very much for the introduction, and thanks to the organisers for inviting me. I've learned so much already today and had some great conversations with people at the coffee breaks and lunch breaks, so it's really a pleasure to be here. So I'm going to be talking about physical activity and the ageing brain. And physical activity, of course, is one of the key modifiable lifestyle factors that we know is associated with successful aging or healthy aging, particularly when we think about brain function. So obviously, diet is the focus of this, the main focus of this symposium in terms of nutrition, but it's only one of the key lifestyle factors that we know alter the risk of developing neurodegenerative diseases and pathology with aging. Other factors include cognitive stimulation, also social interaction. So we know that loneliness is a very important independent risk factor for development of dementia. And this, of course, is very much on our minds when we think about what we've been through in terms of COVID and people shielding and so on. And also physical activity, which is my particular focus of research. So these factors are independently modify the risk of development of dementia and neurodegenerative diseases, but they're also connected. They're very much interconnected, not just at a behavioural level. So we know, for example, that engaging in physical activity, or those who do so habitually, are more likely to have a healthy diet. So not just at the behavioural level, but also at the level of inter-organ communication and signalling. So we've already mentioned throughout today, or various people have mentioned physical activity, and we've identified some of the potential underlying mechanisms underlying why physical activity is important in protection of brain health throughout life. So these include stimulation of expression of growth factors like BDNF in the brain, and of course, BDNF is a trophic factor that is involved in trophic supportive neurons, but also very much involved in synaptic plasticity and learning and memory and so on. Sandrine has given us a fantastic overview of hippocampal neurogenesis, and we know that exercise is a very potent stimulator of adult hippocampal neurogenesis. And also physical activity, of course, very important in protection of the general cardiovascular system in general with age, and particularly then the cerebral vasculature, so decreasing the risk of stroke and of TIAs and so on that are associated with impaired cognitive function with age. But today I'm going to focus on the potential for physical activity to modulate neuroinflammation. Now, Claudio has already given a masterclass on inflammaging, so I'm not going to give another overview of that. But we know, of course, that age is associated with an increase in inflammation in various different tissues, including the brain. And as he has mentioned, this is not necessarily pathological, but it can be, and it can cause damage to various different organs. So things like changes in gene expression, accumulation of cell debris, and then when we think of that in combination with immunosenescence, the inability of immune cells to deal with that accumulated cell debris, mitochondrial dysfunction, and I'll be talking about some metabolic changes and metabolic pathways later on, and microbiome changes, which we'll hear a lot more about tomorrow, I know. So thinking again about the capacity for some of these lifestyle factors to interact, and thinking about how that impacts on inflammation specifically, one hypothesis is when we think about, for example, adipose tissue. So when we have a healthy diet, when we have regular levels of physical activity, we have adipose tissue is in a healthy or a normal state. So it's not acting as a source of pro-inflammatory cytokines and driving the kind of inflammation that is associated with inflammaging. But when we have a situation where we have overnutrition, positive energy balance, and that's combined with a sedentary lifestyle, the resident macrophages within the adipose tissue take on a pro-inflammatory phenotype and function, and they start to act as sources of pro-inflammatory cytokines that will have an impact on various different organs, causing systemic inflammation, and also, as is my interest, causing changes in inflammation within the brain. So when we think about the concept of neuroinflammation, obviously there are specific cells that we need to consider. So we know, of course, in the healthy brain, in the adult brain, neuroglia act as support for neurons, supporting their function, supporting sort of anatomical structure, supporting physiological function, and so on. And we need to think in particular in the context of immune function about microglia, which are, of course, the resident immune cells of the brain. They're the first responders in terms of injury. They respond to PAMPs and DAMPs and will change their function in order to resolve inflammation. But astrocytes, of course, have a very important homeostatic function, not only in providing metabolic support to neurons in terms of the lactate shuttle and so on, and preserving the blood-brain barrier, but also they can contribute to inflammatory responses within the brain. So we need this communication between the different cells of the brain in order to maintain good homeostatic function. And with age or with various types of disease, so this isn't just specific to age, we get some dysfunction of these cells. So in terms of microglia, we can, again, getting back to the concept of accumulation of cell debris, cell senescence, microglia taking on this pro-inflammatory phenotype, and they're not able to carry out their sort of homeostatic surveying function in the same manner as they would in the healthy brain. And astrocytes can become reactive. They have a reduced role to play in general neuronal support, so they're switching to a cell survival type mode, and we get an enhanced immune response. So it is important, of course, that astrocytes and microglia are able to mount an inflammatory response and an immune response, and this is perfectly physiological and it is perfectly homeostatic. And of course they communicate together via soluble factors, they communicate with each other and can regulate each other's function. But with chronic inflammation, inappropriate inflammation, age, disease, and so on, this can switch over into a sort of a maladaptive response, a maladaptive homeostatic response that is associated with cognitive impairment. So we get reactive astrocytes, pro-inflammatory microglia, and so on, creating this inflammatory environment within the brain. And this is where we believe that there is a role for exercise. So exercise might be able to act at different time points and on different cells, perhaps, to reverse or perhaps prevent these inflammatory responses that we see in these kinds of pathological states. So I'm going to present some published and some unpublished data showing how we've tried to interrogate this within my lab. So we started off by asking whether exercise can prevent cognitive impairment that is induced by acute inflammation. So all of the data that I'm going to present to you today has been generated using animal models, using mice. And the exercise modality that we use in the lab is treadmill running. So we use this in preference to wheel running because we want to be able to give a precise dose of exercise to each animal, and we want to be able to house them together. So rather than giving them access to individual wheels, and of course if you group house with a wheel, then you don't have control over which animal is running in the wheel and so on, and how much exercise that they get. So that's the reason why we use treadmills, in order to control the exercise dose. So we habituate mice to the treadmill and we give them 1 week of exercise. This is not very high intensity exercise. It's 30-minute bouts each day. At a sort of a moderate level. And then at the end of that, we challenged the animals systemically with lipopolysaccharide to induce an inflammatory response. We carried out cognitive testing and harvested the tissue for analysis. And first of all, in terms of the cognitive testing, again, generally we use in my lab object exploration type tasks. And this is because they aren't motivated, so we don't want to restrict food or provide a food reward or shock or anything like that. So the animals are perfectly free to explore or not as they wish, the objects that they find in the open field. So the animals are trained to explore objects in the open field. At a later time point then, during testing, in some experiments we swap an object for a new object. This is novel object recognition. Or in the spatial variant of this task, we just move one of the objects. So that's sort of recruiting the hippocampus for spatial learning. So what we found here is that there wasn't any particular preference shown for any particular object during the training phase, but during the testing phase, that animals that were treated with saline, not LPS, but animals that were sedentary, that didn't get exercise. So we divide the animals into 2 cohorts. one cohort that are just put on the stationary treadmill to control for environmental enrichment, but they don't actually exercise because the treadmill doesn't move, and another cohort that do in fact exercise. So we find that these sedentary animals treated with saline are able to recognise the moved object and spend more time exploring it during testing, but that LPS, induction of neuroinflammation, blocks this. So what happens when those animals are exposed to prior exercise, or when another cohort of animals is exposed to prior exercise? This LPS sort of abolition of learning and memory is blocked. So prior exercise is able to protect, in terms of cognitive function, to be able to protect these animals against this LPS-induced impairment. And when we look at what's happening in the tissue, we find that we have a nice, robust inflammatory response induced by LPS. We're looking at expression of IL-1, TNF, and markers of activation of astrocytes and microglia, but that is blocked to a greater or lesser extent, depending on which variable that we're looking at, by prior exercise. This is in hippocampal tissue. We also isolated astrocytes and microglia from the brain to look at the inflammatory profile. And very quickly, just to explain, what we found was that with animals that have an acute inflammatory stimulus with LPS, we get the microglia and the astrocytes adopting a pro-inflammatory state that is associated with cognitive impairment, but prior exercise is able to block that. So we're seeing markers not of the pro-inflammatory response, but of the anti-inflammatory response in astrocytes and microglia. So exercise can protect against this experimentally induced neuroinflammation. Now, thinking in particular about microglia, in recent years we've become quite interested in other methods of assessing activation of microglia. And immunometabolism that field has just exploded over the past number of years. So looking at the metabolic pathways that are adopted by cells in response to pro-inflammatory responses. So in the sort of resting homeostatic state, microglia use oxidative phosphorylation to generate energy. But when they adopt a pro-inflammatory state, the metabolic pathways shift, and they are metabolically reprogrammed. They switch temporarily to aerobic glycolysis. Now, and this is in response to pro-inflammatory stimuli like IL-1 or LPS and so on, to PAMPs and DAMPs. And so we get an increased glycolysis that is characterized also by an increase in the expression of the enzyme PFKFB3, which is sort of the master trigger or regulator of glycolysis, and a decrease in oxidative phosphorylation. Now, this is metabolically inefficient. It produces fewer molecules of ATP when compared with oxidative phosphorylation, but it has the advantage of being much more rapid. So it's enabling a sort of a rapid pro-inflammatory response from these microglia. And then, of course, when the response or when the stimulus is removed and the inflammation is resolved, there's a switch back to oxidative phosphorylation, which is what happens here. So again, in terms of pro-inflammatory responses, the cells are producing pro-inflammatory cytokines and also adopting a phagocytic kind of phenotype in order to clear up any damage or debris. There are, one of the ways of looking at immunometabolism is to use a seahorse analysis, where you're looking at, you can simultaneously measure in cultured cells oxidative phosphorylation and glycolysis by seeding cells into a plate where there's some probes, and you can add in then glucose, or you can add in inhibitors of mitochondrial complexes and so on to look at what's happening in the metabolic pathways. So this is unpublished data that we've produced recently. So we effectively repeated the experiment that I just told you about giving animals exercise, challenging them or not with LPS, and then checking what's happening in the cells. And what we find when we look at the measure of glycolysis here, which is proton efflux rate, we find that the basal glycolytic level in the animals that were treated with LPS is increased. So we're seeing this glycolytic switch in response to LPS, which based on the literature is what we would expect to see. And this is associated with an increase in IL-1 beta. But animals that have been exposed to prior exercise, that glycolytic switch is reduced, it's blunted, and that's associated with a decrease in production of IL-1. So again, we're looking at ways of examining the inflammatory response in these microglia and to see how exercise might be able to interact here. So that's in young, healthy animals. So I've talked already about the fact that astrocytes and microglia are very important in maintaining homeostasis in the brain, and how we can get this sort of dyshomeostasis in the aging brain associated with reactive astrocytes and activated microglia. And so our hypothesis was, if we have some inflammatory damage within the brain, not induced experimentally by LPS, but just with normal aging, are we able to correct that, or to change that, or to treat that by exposing these animals to exercise? So these were experiments that we did in collaboration with Marina Lynch and Kingston Mills in Trinity College Dublin. So effectively, it's a similar type of protocol, but this time we weren't using LPS as a stimulant. We were looking at young animals and aged animals. So the animals underwent treadmill running. We carried out cognitive testing, and we collected tissue. So we isolated microglia to carry out the seahorse analysis to look at metabolic pathways, and also we looked at various other markers of inflammation. So first of all, in terms of the behavior, we did both novel object recognition and object displacement, and you can see here that there's an age-associated impairment in performance of the task, and that is reversed when these animals are exposed to exercise. So these are aged animals that have undergone 1 week of exercise, and were able to show a cognitive improvement as a result of that stimulation. We see that age is associated with an increase in expression of IL-1 beta, so we have got these neuroinflammatory changes within the brain, and again, that is reduced with exercise. And IBA1, which is a marker of microglial activation. Interestingly, well, what we see here is that the age-related increase wasn't present, or wasn't significant, but we see a decrease in IBA1 expression both in the young and the aged animals as a result of exercise. So looking at the metabolic pathways, we see that with the aged animals that weren't exposed to exercise, we see this glycolytic switch present. So the switch to aerobic glycolysis, away from oxidative phosphorylation. So here we're assessing glycolysis, and here oxidative phosphorylation. And that wasn't evident in the animals that were exposed to exercise. So this short period of exercise in these animals is able to reprogram the microglia away from this glycolytic, from being glycolytic to using oxidative phosphorylation. And then looking at some other measures, we assessed expression of PFKFB3, which is this master regulator of glycolysis, and we see the age-related increase that's again blocked by exercise. And when we look at function, not just in terms of the cognitive function, but the phagocytic capacity of the microglia, we saw that with exercise, the animals had an increased, or the microglia prepared from these animals had an increased capacity to phagocytose beads in an in vitro assay. So the number of the microglia that could phagocytose more than 10 beads compares with less than 5 beads was increased. So an increased phagocytic capacity. So our hypothesis to date is that with microglia anyway, that with age, we see a switch in the microglia to these sort of glycolytic pathways, reduction in phagocytosis, and this sort of chronic inflammation, and that this is reversed by exercise. We see the switch back from glycolysis. We see a restoration of the phagocytic ability and resolution of inflammation. Of course, the important thing to think about is that this switch, obviously, to the glycolytic phenotype is metabolically very expensive. It's good in terms of homeostasis, in that it requires it enables this very rapid response, but it can lead to cell senescence and fatigue over time when we have this sort of chronic inflammation. And so this is resolved by exercise. So finally, what we wanted to do was to see, well, exercise appears to be therapeutic in this sort of short-term exposure of aged animals. What about if we exercise animals when they're younger? So what if we have exercise occurring during aging? What will happen then in terms of neuroinflammation? So can we see sort of homeostatic effects of exercise in the aging brain? So here we had, what we decided to do with this cohort of animals was to exercise them or have a group that were sedentary from 3 months old to middle age. So the animals were exercised 3 times per week over a period of months. And we stopped at middle age. And all of the animals were sedentary from that point. So really what we wanted to see was whether early life exercise is protective later in life. So all animals were sedentary up until we sacrificed them at almost 2 years. So we did regular cognitive testing using the object recognition and displacement tasks, and we also gave BRDU injections to check for neurogenesis. So what we found was that exercise was associated through youth through to middle age with an increased performance of the cognitive tasks, and that we see, of course, an age-related decline in the animals that remained sedentary for their entire life. But for those that exercised, even when we stop exercise at this time point, they have an advantage in terms of cognitive performance right the way through to old age. So we wanted to look to see why this might be. And of course, neurogenesis is a possibility that over time we're building up this reserve as a result of increased neurogenesis. And we in fact see this borne out to be true. So the BRDU positive cells in the aged animals, aged sedentary animals, we see an age-related decrease that is prevented by prior exercise, even when that exercise stops at midlife. And we co-labeled with NUN just to check that these were neurons, and we found that was the case. We have an age-related increase in inflammation. So IL-1 and TNF-alpha, pro-inflammatory cytokines, increased expression in the hippocampus. IL-10 is an anti-inflammatory cytokine. We don't see any change with the aged sedentary animals, but with exercise we see an induction of IL-10 here. Again, even when animals haven't been exercised for a long period of time. And then when we isolate microglia and astrocytes, again, we see the pro-inflammatory profile associated with the long-term sedentary behavior being reduced by early life exercise. So to summarize then these results, we've shown that prior exercise blunts experimentally induced inflammation by LPS and protects cognitive function in young animals. That exercise reprograms microglia in older animals, enhancing cognitive function. That lifelong sedentary behavior induces neuroinflammation associated with cognitive impairment, and this can be prevented by earlier life exercise. So in terms of what we're looking at currently and future projects, we're further investigating this metabolic reprogramming of cells by exercise. We're looking at exercise doses and the time frames. Sex difference is very important. I have to hold up my hand and say all of this work was done in male mice, but we're using equal numbers of male and female mice at the moment because, as has been mentioned before, there are sex differences in inflammation as in most other physiological changes. And we're also looking at linking the function of peripheral tissues with central function in the context of exercise. Because we need to think about, well, what is the link between contracting muscle and what's happening in the brain when we exercise at various different time points. So we know that there are a variety of different exerkines produced, not just from contracting muscle, from active muscle, but from other tissues that are active during exercise, for example, adipose tissue and liver and so on. And at the moment, we're working on the hypothesis that muscle-derived lactate during exercise can be taken up into the brain via monocarboxylate transporters and might be directly impacting on microglia, astrocytes and neurons via modulation of metabolic pathways. So I leave it there. I'll just thank the people who contributed to this work, the lab members and our collaborators, Marina, Kingston and Virginia. Thank you.